I welcome you all to the next lecture in our anthropology series. So we were doing human genetics, and today we are going to cover the next topic, that is the karyotype analysis or the cytogenetics. Okay, today we'll do the cytogenetics, and the next one we'll do it's the biochemical methods. Today we are going to cover these two, right? So now karyotyping or cytogenetics. Okay, so it is made, you should understand that it has two words, genetics, and then there's a word called cyto, right? So in this, basically genetics mein we are studying genes. This much you have understood. Genes are a part of, you can say, the chromosomes, है ना? Chromosomes में there are genes, ठीक है? And DNA is basically where all the encoded information. For example, if you are getting any information from your parents, and this information is whether your whatever traits that you're getting, right? Whatever traits, attributes you're getting from your parents, they are encoded in the form of information, and this information is a part of your DNA and genes, right, which are, you know, present in the chromosomes, right? So when we are studying genetics, we cannot study genetics without studying the chromosomes, right? And in chromosomes we basically study two parts. Okay, when we are studying chromosome, any chromosomal study we are doing, we are going to study two parts of a chromosome, the number and its structure. Now I'll tell you how. ठीक है, number and structure we are going to study, and based on this we will say that this is cytogenetics. ठीक है, now I will tell you what is exactly karyotype. Cytogenetics हो गया is the study of your chromosomes, ठीक है, and उसमें we are going to study about the numerical part as well as the structural part of the chromosomes. And karyotyping analysis क्या होता है? Karyotype is basically, it's a way of studying human genetics. Obviously whatever we are doing is a way of studying human genetics only. Abhi tak whatever we did, it was a way of studying human genetics. But karyotype is a descriptive chart, ठीक है, showing the set of chromosomes. जैसे हमने क्या बोला था कि they were Mendel's laws, ठीक है, and when we were trying to show with a picture, right, that was pedigree analysis. हमने pedigree analysis में हमने क्या किया था? We were drawing a picture that, for example, this is the couple, they are having a child who's affected. ठीक है, affected को हम color कर रहे थे, and maybe because the mother was affected. So this is what? This is a pictorial way of showing a thing. इसको देख के you might not understand what it is, but a person who knows pedigree analysis, he will understand, okay, this is the father, this is the mother is affected, and therefore the child is affected. So this, only these three, this and this, these 4-5 symbols show a meaning, right? So it's a pictorial diagram, right? Similarly karyotyping में क्या है? Cytogenetics में क्या है? Cytogenetics में we are studying the chromosomes, ठीक है, chromosomes की detailed study we are doing, and karyotyping is a pictorial diagram of the chromosome. अब इसमें होता क्या है? Basically we photograph the chromosomes and all of that. So इसमें थोड़ा सा photographing होती है. I'll tell you how. Now क्या होता है basically, how will you do it? पहले मैं आपको वो बताती हूँ, because when you understand the procedure, then you will understand, okay, what is karyotyping. So what happens is, we generally take a blood sample, because if we want to study anything, your DNA, your genetic, any information, anything you need to study, you need to take a fluid which contains your DNA, right? And DNA you can take it from any blood, either from a blood sample or from amniotic fluid. It can be, you know, even our hair, our every little thing has DNA, right? So it can be collected through anything, but generally अगर आपको karyotyping करनी है तो उसके लिए आपको blood sample वगैरह चाहिए. DNA तो वैसे हम अलग चीज़ों से भी निकाल सकते हैं, but for a proper karyotyping analysis we use blood samples, amniotic fluid or any other biological sample you can take, ठीक है, which has cells, ठीक है. So you will take the blood sample or any other sample, and this sample will be processed in a laboratory. Obviously, whenever you give a blood sample, you have to go to a lab. They will only do it now. So what they will do is, now they will take your blood samples, they'll separate your chromosomes and stain them, because they have to study certain things. So they are going to stain your or you can say using a dye or a color, you can say, they will stain the chromosome so that it is visible, it is easier to understand, visibly clear है उनको देखने के लिए, right? So after that they are going to scan under microscope for observations, right? So what are they doing? You are giving a blood sample or anything else, they are going to be given to a lab. Lab is now going to separate all the chromosomes, they are going to stain it for better visual understanding, and then they are going to observe under microscope, because these are microscopic things. And then these cell images are photographed. Exactly, they are photographed because you are going to make a karyotype analysis, and I'll tell you what is that. And these photographed images are kept under examination for analysis. Now क्या होता है, I'll just show you with a picture. Now this is a karyotype analysis. I'll tell you now what is this basically. I told you humans have 23 pairs of chromosomes. Now in a karyotype analysis, what they do is they separate different pairs of chromosomes. We have 23 pairs. They put it either in an order. So you can see they are in a you can say descending order. They have been put from large to small size, है ना. ऐसे करके इन्होंने इनको एक systematic arrangement में they have arranged it, and generally this is how a human karyotype will look like. ठीक है. And last one is the sex pair. It is either X, in this case it looks like it is an XX, right, because they both look exactly the same. While X chromosome is a bit longer, Y chromosome is a bit shorter in size than the X chromosome. So this is just a I'm telling you कि XY में जो Y होता है that is a little bit shorter than the X1, right? So this is a karyotype analysis. What they have done is they have taken all the chromosomes, they have separated them, they have stained them under different colors so that it is easier for, because they have taken this information for some reasons. They are studying something, maybe they are studying a genetic disorder. For example, just I'll tell you, देखो, there is a disease called Down syndrome. ठीक है, there is a disease called Down syndrome, and this disease is basically a trisomy 21. Now what is trisomy 21? I'll just tell you for now. I told you chromosomes में whenever we are doing a study of a chromosome, we are going to study two things about chromosomes. One is its number, second is its structure, right? So structuring में problem कैसे हो सकती है? मान लो a chromosome had to be like this, but some mutation happened and the upper part of the chromosome got deleted and the chromosome is left only like this. So this is a structural anomaly. That means this is a mutation, or this, if this happens in a major chromosome or some important chromosome, so what will happen? It will create a genetic disorder. Yes, what will it do? It will create a genetic disorder. And numerical disorder कैसे होगा? For example, there had to be 2 chromosomes. Obviously we have a pair of 23 chromosomes, तो एक pair में 2 chromosomes होंगे, but instead of 2 there were 3 chromosomes in a pair. तो ये क्या हो गया? एक numerical anomaly हो गया. So same is the case in the genetic disorder of Down syndrome, and it is fairly, you can say, in genetic disorders Down syndrome is one of the, you can say, most common ones. Okay, now this I am not covering here because this is in our next chapter, so I am not covering here, just to make you understand कि हम basically अगर हम karyotyping कर रहे हैं, what is basically karyotyping and why we are doing it. We are doing karyotyping analysis because we are studying these kind of anomalies, right? We are studying numerical anomalies, we are studying structural anomalies, and इसमें numerical में trisomy होती है, monosomy होती है, nullisomy होती है. So there are different. It is just one example that I'm telling you. These we are going to study in a lot of detail in the next chapters. So I'm not covering it here, but I'm just telling you these are the things we are covering. ठीक है? Now let me make a karyotype and I'll tell you how. For example, जो okay, let me take this color and let me start making the let me take this color and let me start making the this is okay, let me write first 1 2 3 4 5 6 7. Okay, I will have to take this out. Okay, please bear with me till the time I'm just drawing it for you, because this is important. अगर मैं ये नहीं करूँगी, you will not be able to understand कि what is the concept fully. 17, 18, 19, 20, 21, 22 and 23. Okay, so now let me start to draw them फटाफट से. Now these are a pair of chromosomes that I'm drawing. Fine. Yeah, these are pairs of chromosomes that I'm trying to draw. Now just look very seriously here, because as I told you, I am now showing a karyotype analysis of a patient who has Down syndrome. Okay, so now just try and understand here. Now let me change the color and make it like this. As I told you, in a normal pair, what should have happened? It should have been, okay, let me draw the others also, and this is, ठीक है. Now let just imagine this is a patient of a Down syndrome. Now basically एक normal karyotype में क्या होना चाहिए था? There should have been 23 pairs, so basically 46 chromosomes. But in a case of Down syndrome, there will be an extra chromosome in the chromosome number 21. So what is now we are trying to do is, जैसे एक normal karyotype था, ठीक है, this is a karyotype of a normal person, maybe he has all his chromosomes, ठीक है. So now somebody is thinking that there might be a possibility that this child has certain genetic disorder, so let's get its karyotype done. Now when they photograph, when they separate, they stain the chromosomes, etc. So what they find out is that there is an extra pair of chromosome in the pair number 21. So by photographing also you understand a lot about it. कैसे पता लग गया? ओ आपने देख लिया कि 23rd नंबर 21st नंबर पेयर में there are 3 chromosomes instead of 2. So karyotyping से you can understand you are basically trying to study chromosomes. Similarly, it is also possible that in a karyotype analysis you see that maybe one arm of a chromosome is missing. तो basically normal chromosome क्या होता है? When both the arms ये होती है, जैसे if I say this is P arm, this is Q arm of a chromosome, ठीक है. So in any case, इस केस में P arm मैं बोल रही हूँ, इसकी P arm missing है. तो just by photographing, just by creating a picture view, you can understand a lot about the organism and whether he is healthy or not. ठीक है ना, पिक्चर से ही पता लग गया. We can understand, okay, P arm is missing, so this might be a disease, right? So ऐसे करके you can understand a lot about genetic disorders, right? So karyotype analysis is this. So I hope you've understood it, right? Now these topics you must be thinking, what is this 9.1? Let me tell you, 9.1 is just the gist of what we are going to do from 9.2 to maybe 12th chapter of the or the remaining part, you can say, of the anthropology, because everything now is biological anthropology also only. So do not worry if you're not understanding it, because 9.2, 3, 4 mein humne, we are going to read these things only, and it'll be very clear by the end, by the time we are going to reach to our 12th chapter. So do not worry, just try and understand what I'm telling you right now, right? So now comes the biochemical methods. Now biochemical method mein one of the biggest name is A. Garrett, right? So he tried, what he did, he saw that okay, there are certain diseases that are maybe caused by some enzymes and proteins in our body. So he started to study these enzymes and protein in our body, and based on this he came up that there are certain defects, or you can say genetic disorders, because of that. So isko usne kya bola, and he said that these defects are there from the birth itself, and he called them inborn errors of metabolism. Now what is metabolism? You need to understand, we eat food and that food gives us energy, right? And then the waste material is excreted out of our body. So precisely metabolism is this only, how the body changes the food to energy, right? And then gets rid of the waste. So this is metabolism, and this metabolism, whatever food we eat, it is broken down, ठीक है, उसका breakdown होता है body में through the help of certain enzymes and proteins, right? So these enzymes, like जैसे हमारे bile juice है, there are so many other enzymes in our body that helps us to break down this food, right? So हमारी body में enzymes, protein have a lot of important role to play, and sometimes if there is any anomaly, or there is a certain the enzyme may not be functioning properly, and that also can create certain birth defects, certain diseases, right? It can give rise to diseases, right? And he called it inborn errors of metabolism, right? And because he said that it is present from the birth itself, it is believed in that they are present from the birth effect, so that actually hinders our way of processing or metabolizing, okay? And then it creates disorders. So these disorders can be studied by the biochemical methods, okay? There are hundreds of enzymes and hundreds of problems that can be studied using the biochemical methods. Now there are for example, which disease? So a very famous disease is there, it is called Tay-Sachs disease, okay? It is caused by an enzyme. So mutation in this gene for the Tay-Sachs can cause this disease, okay? So lab test में क्या होता है, what they do is lab test में they can check whether this enzyme is lacking in the people and what creates a Tay-Sachs disease, ठीक है? जैसे for example you can say some person has a Tay-Sachs disease, right? Now you will check their enzymes, ठीक है, which all enzymes are present in the body, which are all are lacking in the body. Based on that you can create a correlation, okay? Maybe you can say enzyme A is missing in this person and he has this Tay-Sachs disease. So you try and correlate maybe enzyme A, because it is lacking, it might be creating the disease Tay-Sachs, right? So based on that you can study enzymes in the body, and then you can study the diseases as well, right? So newborn का screening होता है, ठीक है, you can check, and for this also you need certain fluid. So you can either take a blood sample, you can take any other body fluid, ठीक है, because you need to take enzymes and study them properly, right? So children का कर सकते हैं, ठीक है, 24 to 48 hours after birth, ठीक है, and for उनका biochemical testing कर सकते हैं, that means testing of their enzymes and proteins, and based on that you can see whether the child is healthy or not, if it has any health problems, right? And it can be treated as well, because there are many ways that you can artificially also provide them with those particular enzymes, right? So this way you can protect or cure many diseases. So this is one of the way, and this is one of the studies in the genetic methods. ठीक है? Tomorrow we are going to cover, I think one lecture we'll take tomorrow to cover this chapter, because this is just an introductory chapter. We are going to cover all of this in you can say comprehensively, so do not worry about that. I hope you liked the lecture. If you did, guys, please like, share and subscribe. Any doubts, please post in the comment section below. I'll be happy to answer you. Thank you.